Welcome, students, to today's lecture on histology. Today, we will embark on a fascinating journey through the complex world of neuroanatomy, unraveling the intricate structures and functions of the central nervous system, CNS. Neuroanatomy is a cornerstone of medical education, as it provides a fundamental understanding of the brain and spinal cord. So, let's delve into the world of neurons, glia, and the development of the CNS. Neuroanatomy is the study of the structural organization and functional principles of the nervous system. It encompasses the brain, spinal cord, and their numerous connections. The nervous system acts as the control center of the body, coordinating and regulating various physiological and behavioral processes. The structural organization of the CNS comprises different regions, each specialized in particular functions. These regions include the cerebral cortex, basal ganglia, limbic system, cerebellum, and brainstem. Moreover, the spinal cord serves as a relay center, transmitting sensory information to the brain and coordinating motor responses. Understanding neuroanatomy is crucial for diagnosing and treating neurological disorders. Imaging techniques, such as magnetic resonance imaging, MRI, and computed tomography, CT, allow physicians to visualize abnormalities in brain structure and function. Neurons are the main cells in the nervous system that send electrical signals. They have different parts like dendrites, which receive signals, a cell body with a nucleus, and an axon that sends signals to other cells. Neurons communicate through electrical impulses called action potentials, and also use chemicals called neurotransmitters at synapses to enhance or inhibit signal transmission. Studying this communication is important in understanding diseases like Alzheimer's and Parkinson's. The developmental biology of the CNS encompasses the processes by which the nervous system forms and matures. It begins during embryogenesis when the ectoderm differentiates into the neural plate, giving rise to the neural tube, which ultimately forms the brain and spinal cord. During development, orchestrating factors, such as growth factors and transcription factors, guide cell proliferation, migration, and differentiation. The neural tube differentiates into distinct regions with specific functions, including the forebrain, midbrain, hindbrain, and spinal cord. The precise timing and coordination of events during CNS development are crucial for normal development. Disruptions can lead to significant congenital malformations, such as spina bifida, neural tube defects, and brain abnormalities, highlighting the importance of understanding developmental biology. The meninges are protective membranes surrounding the brain and spinal cord, with three layers: dura mater, arachnoid mater, and pia mater. Cerebrospinal fluid, CSF, between the arachnoid and pia mater serves as a cushion and circulates around the CNS. Disruptions in CSF flow can cause disorders like hydrocephalus and meningitis. In summary, the CNS relies on a specialized vascular network for oxygen and nutrient supply. Major arteries and their branches provide blood to the brain, while a system of veins ensures proper drainage and intracranial pressure. Knowledge of this system is crucial for diagnosing and treating vascular disorders. The central nervous system, CNS, is made up of gray matter and white matter. Gray matter contains neuronal cell bodies, dendrites, and synapses, while white matter consists of myelinated axons. Gray matter processes sensory information and controls motor functions, while white matter facilitates communication between brain regions. Disorders affecting gray and white matter can lead to neurological conditions. Neuroglia, often referred to as glial cells, are non-neuronal cells that support and protect neurons within the CNS. These cells substantially outnumber neurons and play essential roles in maintaining proper neural function. The different types of glial cells include astrocytes, oligodendrocytes, microglia, and ependymal cells. Astrocytes provide structural support, regulate ion and neurotransmitter concentrations, and participate in the blood-brain barrier formation. Oligodendrocytes, in turn, produce myelin, a fatty substance that wraps around axons, providing insulation and enhancing signal transmission efficiency. Microglia are the resident immune cells of the CNS, performing immunological surveillance and phagocytosis. Lastly, ependymal cells line the ventricles and play a role in producing and circulating CSF. The dynamic interactions between neurons and glial cells are critical for normal neural function and can be disrupted in diseases such as gliomas or multiple sclerosis. The spinal cord is a bundle of nerves that extends from the brain to the lower back. It transmits sensory information and coordinates movement. It is made up of gray matter, which contains interneurons, and white matter, which contains ascending and descending tracts. Each segment of the spinal cord gives rise to pairs of spinal nerves that innervate different parts of the body. Understanding the spinal cord is important for treating spinal cord injuries. In the video section on the major divisions of the brain, we explore 4 key regions: the cerebrum, cerebellum, diencephalon, and brainstem. Each region has distinct functions, such as cognition, motor coordination, and vital organ regulation. Understanding these divisions is crucial for comprehending neurological diseases. The ventricles in the brain produce and circulate cerebrospinal fluid, CSF, which cushions the brain and spinal cord and provides nutrients. There are 4 interconnected ventricles, and CSF flows through channels between them. CSF circulates around the brain and spinal cord before being reabsorbed. Understanding CSF dynamics is important for managing related disorders. The brain is divided into regions called functional areas that have specialized functions. Motor areas control voluntary movements, while sensory areas process sensory information. Association areas integrate information and form complex perceptions. Language areas enable language production and understanding. Dysfunction in these areas can cause specific neurological deficits. Neural pathways are complex networks of interconnected neurons that transmit signals between different areas of the nervous system. They are crucial for coordinating physiological processes and enabling complex behaviors. Major pathways include those for voluntary motor control, pain and temperature sensation, proprioception and fine touch sensation, vision, and hearing. Dysfunction or damage to these pathways can cause neurological disorders. In conclusion, the study of histology helps us understand the structure and function of the CNS. This knowledge is important for diagnosing and treating neurological diseases. Keep learning and appreciate the complexity of the nervous system. Let histology be a tool towards better patient care.